Well, hey, by 11. How you doing? Mr. B here. So I'm going to crash into some extra material, I think, for the course, and I've done this in previous years because I think you need to know a little bit more about the vascular tissue, the and the phloem, and how they grow in and how they do transportation. I mean, the story about plants is photosynthesis. So you need to know a few more players in the game. So in this lesson, I'm going to talk about water conducting xylem, and as it turns out, as soon as we get to the angiosperms, we get some pretty neat xylem. Sort of like this first generation xylem, and then there's a what's this new kind of xylem I'm hearing about? These vessel elements. What are they? Well, pretty neat what comes up. Phloem, we'll look at phloem as well, because phloem itself is interesting tissue. It's specialized, what we call parenchyma, but we'll get to all of that as time permits. And we've got nothing but time because we're digital. So, holy smokes, if you get into botany, there's a heck of a lot that you would get to know about these plants. I think the basic thing that you need to understand is it's all about roots, shoots, and leaves. What do I mean? Well, oop, I think I should set that not to do that again. Roots are just exactly what we're talking about here. Everything that's underground. The shoot system, right? Think of that as the stem. And then, of course, we've got the leaves, right? Over here. And our leaves. So it's all about root, shoots, and leaves. There you go. We'll get into the more specifics of what they're talking about here with meristems and terminal buds. Don't worry about it. For now, roots, shoots, and leaves. Now, we've talked about two types of vascular tissue, but one of the things we hadn't mentioned was the generation two, the upgrade, because when you think about it, tracheids, we talked about tracheids when we were talking about ferns. I mean, that's sort of like web generation 1.0. Right now we have the internet generation 2.0 with all the social interactivity going on. When you look at what becomes the heir apparent here, we get what are called vessel elements. And that's when you are going to love, when you get older, you're going to love vessel elements because they are all about hardwood flooring. In fact, if we didn't have them, they're much more reinforced versions of xylem that make cherrywood flooring, um, oak, 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 maple, and choices like that. Just tremendous. Then we talk about phloem. Now, there of course there's generation phloem or generation one of phloem. Yeah, generation phloem. We've got phloem as it starts out, and then we've got to look at it and say, well, what do we have in there? Right? What is this thing you're talking about here called sieve tube elements? And hey, Mr. B, what are companion cells? Much less these things in between the cells themselves. We call these screen plates, these little sieve plates. Now remember that xylem is all about transporting water, and phloem transports photosynthate. What do I mean by photosynthate? Well, what does photosynthesis make? Sugar. And it transports it up and it transports it down. Depends on the needs of the plant. Xylem's got an easier job. It tends to transport water from the roots up into the rest of the plant. So let's get into xylem first. When you look at xylem, and let's pop out of this for just a moment. I'll just turn that off for one sec. Here we go. When you look at xylem, we had been looking at a structure called tracheids. And the tracheids in this section of wood that we're looking at here, in this case, this is walnut. Sure, there's tracheids in a hardwood like walnut here, right? But then you see these large, sort of gaping, cavernous kind of tubes. And that is when you get the vessel elements. That's what these are. And they're pitted like tracheids to allow water to flow in, and it tends to flow up. And they're tremendously reinforced. In fact, the reinforcing around these makes the flooring extremely hard, and walnut is a great choice. Um, you have to be a little careful. Not all hardwoods are created equal. If I was to do flooring, at a very minimum, oh boy, I would be probably going with cherry wood because that is extremely hard. And if you've got pets, things like that, little kids, right? We love them all, but they can leave behind the nail marks, etc. Good news is you can just refinish the flooring. Bad news is that costs a bit of money. If you went with something like pine flooring, here's what would be the problem. Now you notice the difference. You don't see those large vessel elements. And because you don't see the large vessel elements, you're just sort of seeing the tracheids in cross section here, and you can see them running up and down. That's not a vessel element. That's actually a resin canal. Okay, the trees use resin to help them fight infection, for example. Um, the tracheids are pitted, fairly reinforced, but well, I'll tell you what. Just pick up a hammer and tap it down lightly on something like pine. It'll leave a mark behind because these are not as reinforced as the overall structure that you see here. Right? The more vessel elements you have, obviously the more reinforcing you have. They're just much more strengthened with cellulose. It's basically generation 2. And they can conduct a lot of water. Not to say the tracheids can't, but these are something else. So let's pop out for a second here. We need to look at xylem transport. I've got a couple little goodies for you. Now, I'll narrate this because I really want to give you my view on this. Let's move that along. There we go. So I'll just hit stop for a second. Now, underground, the roots absorb the water from the soil, and it passes through the cortex of the root and into something called the xylem. So when you look at the xylem tissue, you see it's very close to this green material here. And this green material, think of that as conducting something else. Let's just look at the blue for now. Okay, that along a little bit. There we go. So the water begins to flow in, it flows into the xylem, and up it goes, passing through tracheids, passing through vessel elements. And that's all good. It's transported upwards, and we'll talk about, well, in this case, I would talk about, you could see the obvious root pressure, and here you see it flowing up because it's got to get to the leaves, and let's just hit pause. What it's done is it's it's flowed into the leaf structure because this is where photosynthesis happens. You need to understand that water needs to be combined with carbon from the air to make CH2O. Now that's a carbohydrate. Specific carbohydrate that will be built up if you give it enough time, C6H12O6. Okay, that's glucose. And photosynthesis happens here in the leaf. Now we're looking at a cross section of a dicot leaf, and this is very important for you to know. But what you can see here, this is a vein, and the water is now going into the leaf, and it's being transported around to support photosynthesis. So there you go. The mesophyll just means the middle of the leaf. Now, interesting little trick just happened there. We'll pause. These little chambers on the bottom, those little guys, are known as guard cells. And the reason what they guard is the leaf from drying out. It's their job to let the leaf breathe, and I'll get into this more later, but they also let some of the water evaporate outwards. And that's called transpiration pressure. And that partly helps pull water up the plant. Think of it as a little releasing vacuum, if you will. As the water is lost, it creates a little bit of microsuction, and that pulls more water along with it and keeps water traveling up the plant. So a neat little animation. I think it shows quite a few things. Remember that the xylem that's working is relatively new xylem. It's not the xylem that grew in towards the center of the stem. That largely would be crushed. Okay, so I think we get that. And there we go. Let's pause. The guard cells, you have to ask what they're guarding. They're guarding a hole here called a stomata. And if it gets a little bit too dry out, they'll squeeze over and try to cover up the hole. But we'll come back to that a little bit later. Don't want to gear in on that too much. I'll set this up. Yeah, that'll be good. We'll leave that. And we'll go back to our material. Don't worry, we'll come back because we still have to talk about phloem. Good old phloem. Not that stuff that you play around with. There is phloem, which is interesting. It's sort of like plasticine. Great stuff to mess around with. No, we're not talking about that kind of phloem. Okay, so we've seen sort of our Xylem generation 2 here with our vessel elements. Ooh, I'm going to have to draw over top of that there. Whereas tracheids were sort of like generation 1. And water tends to flow straight up. Now, water flow, it's passive. The plant doesn't have to put energy into transporting water up the plant. They talk about hydrostatic pressure, the pressure coming from the roots, right? That's part of it, root pressure. And I talked about this in a previous lesson. Generally, the flow of this really heavy material called water is in one direction, and the plant doesn't have to do any work. Now, could you imagine transporting water? If I told you to transport water up onto the roof of your house, it'd be like, well, I'd have to get out a ladder, and then I'd have to put it in a bucket, and I'd have to walk up there, you know, if you weren't allowed to spray it. I'm saying if you had to physically transport it, even up hundreds of feet, right? You'd say, well, I'd have to run some kind of pump. That would take energy. Well, as it turns out, plants have a trick for doing it, and it doesn't require any energy. It just relies upon characteristics of water. Okay, so let's take a little zoom in here. There we go. The pitted trachid. And then the vessel elements, you see how they're stacked up and they kind of have those little plates in between them, right? Lining up wall to wall like that. Perforated end walls is what they call that. Let's see there. Let's see. All right. Now that is not opening up correctly, so I'm just going to go back to my original here and reopen it. These things happen. Maybe the file didn't load completely. There we go. Vascular tissue, water flow. No, it appears to be the file. Let's see if I can get to where we need to go. Okay. Sorry, folks, I got to jump down a little bit. So we know that xylem doesn't just carry water, but there's things dissolved in the water, right? And you can think of some pretty important things for plants: potassium, nitrogen, dissolved nutrients, right? All the best part of things you find in fertilizer. A lot of xylem is just dead at maturity. All that's really left behind that forms the pipe is the cellulose. Think of it as a scaffolding. And it has little pits in it to allow for water flow, but the water tends to flow in one direction. And we should look at that now. Why does water tend to flow in just one direction? A couple reasons for that. We know that the roots are pushing up, and we know that there's a little bit of suction created by evaporation from the leaves. So root pressure and leaf pulling, yeah, those are two reasons. Here you go. It's called the cohesion tension model. And we did talk about this in a previous lesson, but there's no reason not to talk about it now. Okay, so water flowing all the way from there, and it can be up, if you think about a sequoia, hundreds of feet. So coming in through the root hairs, and it gets past a little strip here. There's a waterproof strip called the Casparian strip that doesn't let water backflow out. That's some pretty neat stuff. So once the water begins to enter, right, there's root pressure and there's a little bit of suction as water's leaving from the leaves. And water, the little white bits are hydrogen, and oxygen is represented by the red sort of sphere. So in cohesion and tension, the water particles are attracted a little bit to the edges of the container. And I've said this before, if you put water in a glass or any container, it's adhesive to that container. You can't shake it off. So that keeps it from tumbling back down. So this is just referring to the fact that there's a little bit of suction from the leaves as water is being lost. That's sucking it out a little bit. Sure, I get that. But water itself is sticking to the xylem, And as it sticks to the xylem, it doesn't really want to let go of the edge of the container. Now that's really useful because as it sticks to the edge of the container, it won't fall back down. So it's being pushed up, and the water molecules themselves will stick to each other. They're cohesive, and they'll continue to just move up with the downward pressure, with the pull upwards from the leaves. They'll stick to the edge of the walls adhesively, and then water molecules are sticky with each other. So as your friend, the water molecules being pulled up, he can kind of grab you by the hand and pull you up with him because there is some attraction between water particles. And if you put the root pressure, adhesion, cohesion, and evaporation together, that's all a plant needs to pull up water, which weighs 1 kilogram per liter. That's pretty heavy stuff. Here's a really neat animation of it. So if this is the roots and these are the leaves, comes in through the roots, transported up the xylem, comes out into the leaf, which is this structure here, and it goes out through the stomata. These are the guarding cells, right? They guard, but what they guard is the whole, right, which is the stomate, and the plural for that is stomata. That's pretty neat stuff. Pause. You can watch that. It goes on forever. Okay. So 4 factors: adhesion, cohesion, evaporation, and root pressure. Now the question is, once we get past xylem, and there's some pretty impressive xylem, you have to admit, right? Look at those vessel elements there. And they're ringed around, they're kind of embedded in with tracheids. Get the idea? Here's our softwoods and our hardwoods, right? And I'd referred to this before. The springwood is the tremendous growth when it's, let's see here. Go with that. Recent tools, huh? Springwood runs from here to here. That's the best growth. And the summerwood, right? Well, the summerwood is, it's interesting. I guess you're not really aware that in the summer, that's not a plant's best photosynthetic time. In fact, a lot of its work has already been done with the youngest leaves. So if you want to age a plant, you just have to choose what you want to do. If you want to go from summerwood to summerwood and count the dark rings, be my guest. Or you can go from the light rings to the light rings. You choose. On the left, you have the softwood of a conifer, for example, like pine. And on the right, you have a hardwood. And by hardwood, we could just choose cherry because that's my favorite flooring, so I get to quote it over and over again. Now, phloem, wow, this kind of got messed up. I'm going to go to this slide. Phloem's job is to carry photosynthate down from the leaves to the rest of the plant. Of course, it's got to circulate around and feed the plant. So we can carry it down from the leaves, or the needles, if you will, in a pine tree. But it also has to, in the winter, the plant can't just not feed itself. It's hungry. So it has to take some of its stored sugar, usually sugar is stored in the form of starch, and then convert it and transport it up the plant. So how is that done? What kind of a pump do we have for that? Because we're not talking about water right now. We're talking about sort of a sugary, watery material. Like when we were looking at water transportation, that was one direction only. But in phloem, as it turns out, it can go up and it can go down. So what exactly is the mechanism for that? Well, plants, as it turns out, have these neat little things for their phloem. First of all, the pipes that you're looking at, these pipes here that stack end to end, these food conducting pipes are very close to xylem. Now, phloem, we basically are stacked what we call parenchyma cells. And these cells are able to transport sugar up and down. Now, it's not actually what we call the sieve tubes. In this case, this type of phloem is made up of chunks, and we call the these sieve tube elements. And pretty neat. They're alive at maturity. They're not hollowed out and dead like xylem is. And they've got little helpers. And they got these little fellows on the side called companion cells. And companion cells, depending on which direction the sugar needs to go, can run clockwise or counterclockwise. At least that's the best data we have right now. It's believed that there's sort of a swirling of the cytoplasm to help create sort of little micro pumps, depending which way you need to transport your photosynthate, up or down. So I've got a neat little animation of that for you because I think you needed a visual. I know I would. So what have we got? Well, here it is. You've got xylem here where water's flowing, and we kind of get the idea. If we look at a source of sugar, let's say we hook this up to a leaf cell, which is doing the photosynthesizing. Now, here's these interesting little plates, which kind of mark off each cell. The photosynthate can move up and down. Now, let's see how that works. Down here, let's say that we've got fruit hooked up. And then what we want to do is we want to get the sugar from the leaf down into the fruit, right? Because remember, reproductively, we want to create fruit to help distribute our seeds. We want to get animals interested so that they'll help us, the angiosperms, reproduce. All right, so here you go. Pretty used to seeing this. Water going up in the xylem, and it can diffuse across as it needs to, right? It's got to get to a lot of cells to get the idea. Water's needed throughout the plant. So that's pretty par for the course. Now, when we look at these companion cells, they have this impressive job of pumping photosynthate up or down. Okay? You'll see as this begins to rise, of course, water is going to diffuse out to all the cells that need it. Okay, so I'm going to speed this along a little bit. There we go. So now what we've got is the sunlight's on and we're producing sugar, and the companion cell helps to transfer it over. And as the sugar begins to flow, in this case, what the plant wants to do is it wants to get that sugar and some of that water that it needs. And the companion cell, like a little pump, is not only handling the exchange, but it's also helping to get what's needed to the developing fruit cells. So we'll see that some of the sugar will be diverted, picked up, pulled over, and funneled into, well, let's call this an apple, for example. This is going on with my apple trees right now. The fruit itself only helps the plant with reproduction. So my apple trees flowered very early in the spring, and it's been taking a very long time for the apples to develop because what you're seeing here is a sped up process of just how long it takes that sugar to help nourish those fruit cells and begin to build them up. And that's what photosynthesis is really all about. And there you have it. Okay, so let's pop back for a second. And there we go. Realize that it's the companion cells that do a lot of this work. They're the sugar transporters. And I really like this shot because it shows a great number of things together. You've got tracheids, which are xylem, right? And I don't think they haven't given us, in this case, it's too bad, they didn't give us any vessels. But in this case, they did give you the phloem, right? So you've got these large vessels running up and down. And you can see there's sieve plates. And then there's the companion cells right there, adjacent to the, basically the phloem parenchyma cells. There we go. And here's another neat sort of look at it, where you can see, sure, the xylem vessels or the tracheids or whatever you want to call them, right? They're just pipes transferring water, whereas you can see the transport of photosynthate can be up or down. More or less is the idea. Here's a really good shot of it. And this mirrors what we were just looking at in our animation. You could see the diversion of sugar into the apple. That's why it takes apples so long to develop. And water has to transfer from the xylem through osmosis. It's not just a simple matter of getting into the phloem cells. Remember, they're living. So if water has to diffuse to get into living cells, remember, diffusion of water through a membrane is called, gotta remember that, osmosis. Water, membrane, you get the idea. Okay, so that's the end of that little presentation there. It's too bad that I had those blanks in there. Okay, so I'll pop out of this. And the last thing that I wanted to show you was a little bit of information about plant growth. Now, this is kind of important to understand. If you want a plant to grow up quite tall, you have to realize the buds on the plant. There are vertical buds, and then there are buds that go off to the side, lateral buds. Okay, so let's look at a plant growth pattern. If we're going to talk about xylem and phloem, let's just get it all out in the open. Okay, so at the top what we have is something called an apical meristem. Now that's easy to remember because the apex is, let's just back that up for a second. The apex is the top. Now you can have apical meristems. They lead vertically. Okay, so you can have a chief one up here, and you can have them on the root tips as well. Just going to move this along a little bit. And you'll see that the plant will grow in height. Now, if a plant only grew up in height, it'd fall over. It needs to laterally become bigger. So what you get in addition is you get lateral growth. Now, these are called lateral meristems. And there's a little trick. If you want your plant to be taller, you can cut a few of the lateral meristems. Think about the tips of branches, for example. If you cut those, the plant will probably put more of its energy into height. If you're worried about the plant, it's too concerned, it's getting too tall, you can nip a few of the apical meristems, the tips of branches, and slow it down a little bit. Right? That's just called pruning. Widening, though, occurs at something called the secondary meristem. So let's look at what that really means. And I've referred to this earlier on in the lecture. When you look at dicots, they're pretty smart because they have a little layer of what's called vascular cambium. It's that ring. And it's the cells that can become anything. So here comes the secret. This is why something like corn can't get this heavily reinforced xylem. Corn can't form a log. It just never will because it can't form rings of tissue. Now watch as this develops. Sure, we have the pith and the cortex is outside here, and the epidermis is the skin, the bark goes over that, we get it. What I really want to focus in on is this right here. This layer of cells are called the meristematic cells, right here. And they can become xylem or they can become phloem. Now see how they're arranged. The xylem is pointing to the outside, and the phloem is in an organized fashion pointing to the inside. When you look at something like corn, sure, it might have its little vascular bundles, its veins, like this, but they're not all pointing in towards the center. They can be pointing in, out, left, right. They're just completely scattered. And if you don't organize, you'll never get your xylem pointing towards the inside. Every one of these bundles, if it makes xylem to the inside, you get rings of xylem. Corn can't do that. So here we go. The meristem right here, this meristematic tissue is renewed, right? It grows too. But you start out with your primary vascular bundle, and there you can see it dividing. And what it's done is it's laid down some new xylem. And conversely, it lays down some new phloem towards the outside. And what it keeps doing, the youngest xylem, the original xylem is going to grow in towards the center, eventually get crushed, and let's just say primary, I think that's a better term. The primary xylem is being pushed into the center, and the primary phloem is being pushed out towards the edge of the bark. Well, I shouldn't say the edge of the bark. What I should really say is out into the cortex. Now, let's just pause that so you can see what this layer of cells begins to do. Interestingly enough, there is another layer, another little bundle called another cambium that's called cork cambium, and this process goes on outside here. And it's more than you really have to talk about in the course, but it produces tissue as well, and it's another cambium. Let's just let this run for a second. We have our xylem and our phloem. If you didn't have another type of cambium, then you wouldn't be producing. Let's just pause that. There we go. You wouldn't be renewing the structures that are out here. The cork that's needed to go just underneath the bark to protect the epidermis. So there's another layer of protective cells that are growing in a ring too. But I really just wanted to focus in on these rings of xylem. And your primary xylem from year one was here. And if you think of as this grows out, right, your original xylem, or sorry, rather your original phloem, just gets pushed out and it loses its functionality, right? But our vascular cambium, which is renewing our veins, xylem and phloem, is right here, and it just keeps adding to the growth of the tree. It keeps growing as well, but it's not that far away from the epidermis of the plant. And you have to be really careful because if you put a bit of wire around a tree, and if that wire cut through the cambium, you would essentially kill your plant. That'd be it. Just have to pause for a second. Well, that happens sometimes. The phone rings in the middle of your presentation. It's okay. It's around 5 o'clock. Um, Anyways, to make a long story short, the vascular cambium, pretty important structure, renews your xylem, renews your phloem. So I'll let that go. We saw this guy here. He's done. And we should talk about photosynthesis, but I'm going to leave this topic for when I discuss the leaves. Tremendous topic. It comes up. It comes up in your first year of college university studies, but it's not really until you get to biochemistry where you see this in particular detail. But as promised, I'll bring this topic back up when we talk about leaves. Okay, folks, have a good one. Mr. B, out.